Hello everyone, today we are going to learn about lactose operon or Lac Operon. Bacteria use regulatory proteins to turn groups of genes on and off in response to diverse environmental signals, allowing them to adapt changes in the environment. The Nobel Prize was awarded to Francois Jacques and Jacques Monod for their work that improved our understanding of how lactose metabolizing genes in E. coli are regulated. Lactose metabolism in E. coli is governed by 3 structural protein-coding genes. The Lac Operon is made up of these 3 Lac genes. An operon consists of a group of genes as well as a promoter and an operator that control the transcription of those genes. A single mRNA is created when the structural genes in an operon are transcribed. Because it contains information for more than one type of protein, this mRNA is called polycistronic. Lactose is broken down into glucose and galactose by beta-galactosidase, which is encoded by Lac Z gene. Lactose permease, encoded by Lac Y gene, transfers lactose into cells. Lac A gene codes for transacetylase, a protein whose function is unknown. The operator Lac O is a small piece of DNA that is partially within the promoter and interacts with a regulatory protein that regulates the operon's transcription. Lac I gene is a regulatory gene that produces a mRNA that is used to make a repressor protein which can bind to the Lac Operon's operator. A regulatory protein is the general word for the product of a regulatory gene. Because it prevents RNA polymerase from transcribing structural gene, the Lac regulatory protein is known as a repressor. The Lac repressor binds to the operator in the absence of lactose and prevents RNA polymerase from transcribing the Lac genes. Allolactose binds to the Lac repressor protein, altering its structure and preventing it from binding to the Lac operator, allowing the Lac genes to be expressed. RNA polymerase can then bind to the promoter and transcribe the Lac genes. Jacques and Monod created mutations in the Lac operon to demonstrate how they might impact the gene expression regulation. They induced a mutation in the Lac I gene that resulted in the production of mutant, inactive repressor proteins. The proteins are unable to interact with the operator. As a result, the structural genes are produced constitutively, that is, in the presence or absence of lactose. Mutations in the operator are called constitutive or Lac OC. The repressor protein is unable to recognize this sequence due to base pair changes in the operator region. The structural genes are constitutively expressed in the absence or presence of lactose because the Lac repressor cannot bind. Lac IS, the superrepressor, a mutation in the Lac I gene resulted in no Lac enzyme production in the presence or absence of lactose. This mutant repressor protein recognizes the operator but not allolactose. As a result, even in the presence of allolactose, the Lac repressor attaches to the operator, preventing the transcription. The Lac Operon is an example of how bacteria can respond to environmental changes by turning on and off genes. Lactose stimulates the production of enzymes required for the conversion of lactose to glucose. The operon's mutations show how the various regions are controlled.